The overall goal of this procedure is to develop a technology based on Mesoporous Silica thin film for the selective recovery of low molecular weight proteins and peptides from human serum. This is accomplished by first fabricating the Mesoporous Silica thin film chips and pre-treating with oxygen plasma ashing. The second step is to perform a simple pre-treatment of the serum and then to enrich the low molecular weight proteins and peptides from the serum by on-chip fractionation. Following elution of the low molecular weight proteins and peptides from the chip, the molecules are profiled using matrix-assisted laser desorption ionization time-of-flight mass spectrometry. The final step is to utilize professional software to perform data analysis. Ultimately, the mass spectra and statistic analysis results reveal the specificity and efficacy of low molecular weight proteome harvesting.
The main advantage of these techniques over existing technology like 2D page Western blot include the powerful capacity to enrich the low abundant, low molecular weight proteins and peptides in serum, the high throughput and time efficiency, the reduction of the serum sample requirements, and the low cost. One of the most challenging aspects of this procedure is the control on nanopore morphology, such as pore size distribution, pore shape, and pore interconnectivity, because this feature determines the sensitivity and the specificity of profiling target blot signature. To ensure success, we aim to carefully control the molar